thank you everybody for logging on. My name is Udis Sarkans, and so I will present BioStudies today and talk about organizing data associated with the publication using BioStudies. So that's a rough overview of the talk. So after some introductory thoughts, I would like to talk about motivation first. What are the problems that we are solving? Then I will show and talk about practical usage of BioStudies, starting from data access and then following by data submission, so depositions, and we'll finish by talking about some could be called advanced use and also future ideas, future developments. Very quickly about myself, I have a computer science background from the University of Latvia, which is sometimes quite a cold place. But I've been at European Bioinformatics Institute since year 2000. I run the functional genomics software development team. Our main project is the BioStudies database, so both developing and running it. But then we also maintain software for Express, which is a gene expression functional genomics database. And on the data access side, also support users. And more recently, we've been helping with starting up the Bioimage Archive project. So I will return to that later in this webinar. And to introduce the team, so I will have also the names later, but for now only the faces. And instead of an org chart or anything like that, I like to use this view where it's a two-dimensional matrix where we have these main responsibilities of the team versus the software components that we build and maintain, and of course people have to be flexible and move around in this two-dimensional space. Okay, so that's a quick intro. So, well, data heterogeneity in life sciences. Of course, in life sciences, more and more data is being generated, and it's great, and lots of useful insight is found due to the large range of various technologies and approaches. But then that also creates certain problems from the data sharing perspective. So for structured data, where the community databases exist for sequence data sharing, for proteomics, protein structure, and so on, so that's all fine. But then there are more and more what are sometimes called multiomics studies that comprise multiple data types, and so there is one of the aims of BioStudies is to create an umbrella data entity that points to various components of data in individual community databases. Well, and then what is often called unstructured data, it's I don't particularly like this term, and of course the data should always have some structure. It's just that structure is sort of not defined in advance. It's sort of could be called an ad hoc structure. Some Excel sheets, supplementary materials attached to a paper, and so on and so forth. But then, well, there are also cases of data which clearly has a structure, but that structure has not yet been sort of explicitly defined and agreed on by the community. There are no standards. And an example here is light microscopy data, where there are some standards for aspects of light microscopy data, but there is nothing at the moment that covers the entire study. And then finally, the point about supplementary data attached to publications. So even if a publication is open access and the supplementary data files are available in principle, different publishers offer very different access patterns for the supplementary data files. So that creates another dimension of heterogeneity. So all of these are points that BioStudies tries to address. Well, if we look just very quickly at the paper, this is about coronavirus, so quite a recent paper. So we might want to know what data associated to this paper is present in various community databases. Are all the associations to those data sets of the same nature? For example, has the paper created new data sets, or is the paper reusing data from already existing data? And what other data are attached to this paper? So, of course, if we read the paper, then we can find out that the paper talks about five proteins, and the paper mentions these protein structures in protein data bank, since it's about protein structures. So here are the accession numbers about of these 5 proteins. And there is also a reference to supplementary figures. So there is more data associated with the paper, but it's all sort of scattered throughout the text of this article. So then BioStudies. So the idea of BioStudies is to aggregate all research study data. And why? It's really to facilitate transparency and reproducibility of research. So these are the topics that are, of course, recently becoming more and more important with, for example, fair data movement. And the diagram on the right shows the main concepts of BioStudies. So what we call a data package, the 3 main elements are links to data components, either in EBI resources or external resources. Then what could be called orphan data files, so some Word documents, PDF files, TIFF images, and finally metadata that describes the study as a whole, as well as the individual components of the study, and the link back to publication in Europe PMC database. So that's the main idea. And now if we look at that example paper, so if we look at it in the Europe PMC database, then there is a data tab, and then clicking on the data tab brings us to the corresponding entry in the BioStudies database, which has these supplementary, well, there's just a single supplementary data file here, as well as 5 entries, 5 links to PDB. So this is a very, very simple example, but there will be more complicated examples of many more data files and many types of links present in a particular data package. That shows one way how BioStudies acquires data in collaboration with the Europe PMC team after the paper has been already published. But then if we look more generally across the research life cycle, so what are the stages in research life cycle? So of course data is being generated, and we work, we use BioStudies to support various projects while the data is being generated, in the data sharing, and eventually transitioning to publication capability, as well as we sometimes help an emerging community. For example, a protein design data collection so protein design community is thinking about data sharing and creating standards for this kind of data. So that's also where BioStudies can help. And then at the stage when the paper is being prepared, then of course the associated data needs to be published and made available. And then BioStudies can support the authors with publishing the data that does not have a dedicated community archive, And I will talk about both of these examples, bioimage and RxPress, later in more detail, as well as linking to making these sort of links, linking together data components in community archives, and in general providing means to deposit orphan data files that could also go into supplementary materials. Well, and then of course this is a very sort of significant event. The paper is published, and then post-publication we ingest data from your PMC that makes up the bulk, by far the bulk of the BioStudies contents at the moment. And we also collaborate with the Source Data Project. I will talk about that briefly a bit later. Finally, if there are data resources that are being retired, then BioStudies can help with taking the data and continuing the distribution of those data sets. So various categories here, various data sources for BioStudies, but the main sort of one of the principles is that the earlier the data is being thought about, the better it is for reproducibility of science, for open science, for fair data. And yeah, ideally the data package is created before publication. So in that case, if that has been done, and then the data package is in BioStudies, the paper is in the journal, well, it's published, and then a bidirectional link can be created between the paper and the data package in BioStudies. So that's quite a long introduction, but I thought it would make sense to sort of talk about the principles of what we do. Now, if we look at practical, well, data access and data submissions, so BioStudies is available at EBI/BioStudies. The browse view is, so this is all the data sets currently there. It's almost 2 million data sets in total. And there are facets and filters on the left-hand side of the screen. As you will see, most of the bulk of data, as I mentioned already, come from your PMC. And then there are other facets as well that help with finding the interesting data sets. It's possible to sort by various attributes. And then scrolling down, other facets that we support, types of links and types of files that sometimes can be useful when trying to locate a set of data sets. Now, looking at the structure of a single data set, so it's the general principle is that the metadata study annotation is on the left-hand side of the screen, and files and links are on the right-hand side. And the files panel can be, files and links panel can be expanded in various ways here. So this example is a simpler, again, an example of a data set ingested from your PMC, where the metadata is relatively simple. We have authors, we have abstract, and that's it, more or less. However, the model of BioStudies allows much more sophisticated representation of metadata, if that is appropriate. So this is an example of an imaging study where we have a much larger number of data files. So that's one thing, and then we have metadata that is relevant for imaging, such as experiment size, imaging method, and control description. So there is no single sort of the fields in the metadata panel in BioStudies are not predefined. They are sort of flexible and they can vary from study to study. One more example, so the source data project, so that I already mentioned. So the source data, the idea is, it's an initiative by EMBO journals, and the idea is to make the data behind the figures available. So if sort of that it's not enough to just make a figure in PDF available for the research community. It is much better for data reuse to also make the underlying data files available. And the other thing, what they do is they curate each individual figure and identify entities that are present in those figures and describe those entities by pointing to descriptions of, for example, sequences of proteins in various databases. And then this data package is again sent to BioStudies, and we make it available. And then, for example, this biological entity annotation is made available via this table that we have for each figure annotated by the source data team in this way. So lots of heterogeneity when describing different types of studies. Well, queries should be more or less intuitive. The search field, but something that I should briefly explain is that queries in BioStudies are ontology enriched. So here the query would return and color in a certain way also more specific terms. How does the BioStudies know what are more specific terms? So we use the experimental factor ontology, which is a project at EBI that builds a sort of ontology describing experimental variables that are available in various EBI databases, and EFO essentially integrates various other ontologies into a single overall structure, and we use the services of this ontology. Returning to queries, it's possible to combine search terms using capitalized and/or not and brackets. This particular query brought up a synonym, so this is a C lamprey is a synonym of simply lamprey according to the EFO ontology. Another example with brackets, and also to note that or can be skipped, and if there is a longer phrase, then it should be enclosed in double quotes. So these are all the principles that are, I think, in most databases present. So there shouldn't be any surprises here. And then finally if we look at a specific collection of data, for example, all the data sets imported from Europe PMC, then some additional search facets, if they are configured in the BioStudies system, can become available. So for your PMC collection, it's the funding agency, and so it's another criteria that can be easily used when looking for data. And then the final topic on the data access side, data downloads. So this is again a much larger data set with it's again imaging data set with almost 35,000 individual data files. Each of the files is described with additional metadata. So and that additional metadata is searchable. So you can sort and search in these individual columns to narrow down to find a specific image or specific set of images. And then for downloads, if you just want to download 2 images for some reason, after clicking download 2 files, you will see another window which will offer different download methods. So if it's a small set of files, then a download as a zip file, that's fine. For larger downloads, of course, that will not work. If a total data set is a terabyte, for example, then that will not work. Then you would need to use FTP or Raspberry protocols. And the BioStudies will help you by generating an FTP or Raspberry download script, sort of that would instruct to download only the files that you selected in the window. So that's all about data access. Now let's go to data submissions. We have a data submission tool. The main principles, so it enables flexible annotation of a data set as well as associated files and links. It's done via web form, but also there is a tab delimited file upload functionality, and in fact also JSON or XML for use by more advanced users who would like to set up pipelines for data upload. And we provide configurable templates for different domains or projects or collections of data. Well, registration of, you have to register for an account. One thing to mention here is ORCID. So there is an integration with the ORCID system. So you can associate an ORCID to your account, and then it will be available in your data submissions. Then once logged in, perhaps it's better to start with the just list of my data files discussion. It's your private area in BioStudies. So these files are not available to anybody else. It's only for you. They are not associated to any data package or study. They are just being uploaded, you can also upload the entire directories, and you can you can upload via the web interface. But if needed for large volumes, there is again FTP or Aspera mechanisms. So you will get instructions how to use FTP or Aspera for uploads. Each account gets what we call a secret directory, which I have pixelated out because that was my personal secret directory. And then you know how to upload data into your BioStudies account. But then talking about the actual submissions, this is the list of submissions list view. There are two tabs here. On the first one is these are the submissions that have been already submitted into the BioStudies database with the associated release dates. So you can set a release date in the future. We currently do not limit the sort of the furthest away date that you can set this release date to. I think most of the community databases do limit this, so I think it's likely that we will do that as well in future, but currently we do not. And then the drafts, the other tab here will show you submissions that have been initiated but have not yet been sent to the backend, to the database. So these are still in process. And then if you push new submission, then you will be able to annotate your data set. So there will be various fields here, and then the validation messages appear on the left. Once there are no validation errors, then at that point you can submit. Then the system thinks that everything is in order, and you can choose the release date here. Something I did not mention earlier on the data access topic. If you need to provide, if you have a private data set and you need to give a pointer to that data set to a journal or to a reviewer, it's possible. There will be a sort of a secret link that you can share, and people can only access that single data set via that secret link. Scrolling down. So what else is in this metadata template? So contacts, it's possible to add more columns in the contacts list if that's relevant. And then, yes, links and files, as we've been already talking about these a lot. One thing to mention about links is that we use the services of identifiers.org that help users to enter a link to a community resource in the shape of what are called compact identifiers. So it's always possible to have a full URL here. But for example, if your data set is in array express, once you start typing in these first characters, then the BioStudies will prompt you for the possible resources that you might want to refer to, and it will also help you to format your accession number in the correct way. Just to briefly mention what identifiers.org is. So that's an identifier resolution service. Well, there is a long-standing tradition in life sciences to use these compact identifiers in the form of prefix column accession number. Although these days there is more and more demand for DOIs, for example, I think that the compact identifiers will be around still for a long time, and this is a service which is also built and maintained at EBI that helps users to resolve identifiers. Something I did not mention when I talked about creating a new submission. So there is a concept of a submission template that we have. Most users will have only the default and bioimages templates currently available. So let's look briefly at the bioimages template. So the idea here is that if you have a light microscopy imaging submission, then it's better that you provide a little bit more information so that your data set would be truly useful for downstream consumption. And well, the web form, of course, will prompt you for the fields that are relevant here. There will be some drop downs. You can enter your own values if none of these are applicable for your case. And most of the fields are also optional, so it's not that much more work compared to the default template. And this is aligned with the work of our colleagues at the University of Dundee, building IDR image data resource, which is one of resources existing in the world that allows users to publish their imaging studies. One of the concepts here is a concept of a file list. So that in the default template, it was possible to attach individual files to your submission. Well, in the imaging study, if you have 10,000 images or maybe 1 million images, that's not really practical. So the file list enables you to list your individual image files as well as to describe them in more detail. This might be an example of a file list where first column needs to locate to refer to a particular image in your BioStudies home directory that you have already uploaded via one of the mechanisms that I described earlier, and then you can describe sort of any attributes. Defined attributes that help understand what the particular image means. This is all very free form at the moment. As the image data sharing recommendations develop further, we expect that there will be sort of more examples and more more guidance how these file lists should be formatted. Right now there is lots of freedom. So this is the last large section of the webinar, advanced use and what happens in future. I briefly mentioned that it is possible to use, instead of a web form, to use tab delimited files or XML or JSON files for uploading your metadata. It is useful for the cases where you have many similarly structured data sets either at the same time or perhaps over time. It's well, the language of BioStudies is quite simple, we hope. It's described in the submission instructions, and just to briefly show an example of that's one of the imaging data sets. It's a tab delimited format that and BioStudies recognizes some of these headings as keywords, for example, author or publication or link. So the BioStudies will know how to treat a particular element of information. And there are, of course, lots of these files can be, for each of the studies available in BioStudies. These can be downloaded, and so you can look for examples, for inspiration. So I have been talking quite a lot already about imaging data, especially image data submissions. So this is done to advertise a new resource at EBI called Bioimage Archive. It was announced less than 2 years ago. The idea is to create an archive for all kinds of imaging studies that would enable the community to submit data. And of course, we depend on community to submit data, otherwise this will be empty and useless. And then distribute the data, but either directly from the archive, or perhaps more significantly, create what we call added value databases. So enable creation of added value databases that are specific for a particular project or type of imaging, and that can rely on the data, on raw data being in the Bioimage Archive, and then they can access data from the Bioimage Archive and build on top of that additional functionality, nice visualizations and things like that. So that's the overall idea. So how is that relevant for BioStudies? So well, BioStudies is helping this initiative in this sort of pilot, during the pilot phase. We have, for example, built a Bioimage Archive dataset index using essentially BioStudies, just with a different header, where it's possible. In this example, we have queried by this keyword, and we are getting a few studies. Some of them are in Empire, which is electron microscopy database, which is another resource at EBI. And then one of the datasets is in BioStudies. And then, well, once users have submitted the data using what I showed earlier, then for data access, so this just shows another data set in BioStudies. Here, almost 100,000 files. It's a cellular phenotyping experiment where each of the images has a vector that describes various cellular phenotypes. And yet another example, it's a COVID SARS-CoV-2 drug repurposing data set. Again, lots of images and links to drug structures. So these are examples of how BioStudies is used for image data distribution. So briefly, data sharing in a project. So that was the first column on the left-hand side in that life cycle slide that I was showing. It is possible to use BioStudies for sharing and easy data publishing at some point in future in a project, in an ongoing project. So essentially that means setting up a new collection in BioStudies, then designing useful data and metadata formats and managing access rights. So I should note that this cannot currently be done without involving us, but it's something that can be, that we can, if there is interest, we can discuss that. So I will not. I think I've been talking for quite a long time already. I will not go into details of this. So it's just again, considerations about the individual elements of a data set that we've been talking about a lot. So metadata attributes, files and links, and then sections for creating some hierarchy for a more complex data set. And essentially, BioStudies enables sort of designing and trying in practice your own data sharing form. And then, yes, the data access rights can be organized so that individual accounts can submit data into a particular project collection, as well as edit their own data sets. Then there could be a read-only user that can access all the project data, which is not publicly available initially. And then as papers associated to the project are getting published, then the data sets can be released one by one or in larger chunks. And then there is a public access. And I think I will also, in the interest of time, skip over this. So it's really, again, repeating what we have been talking about, the generic nature of BioStudies and the usability of different services that are useful for life sciences data sharing. So the final point is ArrayExpress. So that's a database that you might be familiar with. So it's been around for 20 years. It started out as a gene expression database, but then was later, as sequencing-based technologies evolved, the ArrayExpress was also sort of modified to be able to deal with more modern technologies. So we have in ArrayExpress we have 75,000 datasets collected over time, and something that we realized after we built BioStudies was that that all the services that we offer in BioStudies actually are sufficient, so that we do not need a separate ArrayExpress database and ArrayExpress data access interface. So we are in the process of migrating all the ArrayExpress data to BioStudies. At the moment, you will see a very small portion of ArrayExpress data in the publicly without logging in, but this is ongoing. So this is just to illustrate how ArrayExpress data collection will look in BioStudies. And then looking at a single data set, so that's the current ArrayExpress representation, and we have mapped that into the BioStudies language, and we are getting sort of almost as good result. In some sense, it's better because now we have more flexibility to change in response to user needs what can be changed. So this is in process, and we expect to finish this in the coming month. I should add here that this is only for data access. So for data submissions should still the gene expression data submissions should still be go through the annotate tool, which is a separate tool to BioStudies. Very final slide. So future, well, of course, we want to facilitate both data depositions and data access. So for data depositions, DOI assignments is something that is frequently asked. We can assign DOIs on demand, but it's currently a manual process, which is clearly not scalable. So we are working on a clean treatment of dataset versions. And DOI, well, still on demand, but users will be able just to tick in the submission form, please, I need a DOI, and we will be able to do that. Integration into publishers' pipelines doesn't necessarily mean that the sort of a technical integration. It's about raising awareness of BioStudies as a possible target for data set submissions, and we are also talking with other generic data repositories such as Dryad and Zenodo sort of how to collaborate. And then on the use side, we would like to help measure impact of data sharing, and there are some ideas there. So then help with understanding what is in BioStudies, sort of how, for example, extracting cross-reference graphs from BioStudies. That's one of the ideas. We understand that not everything is, as we had to build quite a generic database, not everything is necessarily very user-friendly. So we need to do more usability studies to understand what users need and where users get confused. And finally, for cloud access, that's clearly also a huge topic. So it's especially for imaging data sets that are large and where it's not practical to download large terabyte-sized data sets. So we are working on enabling. At this point, the idea is to have a Jupyter notebook-based system that would run next to BioStudies on EBI cloud and enable data analysis on the contents of BioStudies and the bioimage archive. So just to acknowledge again the team with the names this time and the main funding sources, as well as the two external projects that I mentioned in this particular presentation, IDR and source data, and we collaborate with many teams at EBI that help us. So thank you for listening. So that's BioStudies URL. There is a help mailing list, and I'm very happy to take any questions.
Thank you, Avash. Thank you, Eugis. Thank you very much for today's webinar, and thank you all for attending today's webinar. So before we go on to Q&A session, Eugis, could you go on to the next slide?
Yes.
Yeah, great. Yeah, so just to let you know that this webinar was part of our A Guide to Webinar series, where we are telling you about concepts in bioinformatics and how you can discover different EMBL-EBI resources through this webinar series. And at the end of this webinar, when you leave the session, there will be a pop-up survey for sharing your feedback for this webinar. So please share it with us. All right. So now with that, we can go on to the Q&A session. So we have Avais from BioStudies who has joined us. He has already answered several questions, but there are still some open questions we have. So I can read some of those. So one question we have.
Yeah, please.
Yeah. So, or if you'd like to read it by yourself.
Yeah, no, I can read myself. So, so can multiomics data present in a metadata study be submitted to BioStudies? Yes, absolutely. So, well, BioStudies is the only sort of restriction with BioStudies is that if the data is sort of if it's human identifiable, then that's not something that we have mechanisms for dealing with. So if the data is granular and needs, for example, some data access committee for essentially if it cannot be public without any logging in, then BioStudies is not the right target. So the next question, what type of data is not suited for BioStudies and should be uploaded on other databases? Or would you recommend to upload, link everything there, also structured data? Yes, so BioStudies should not be used for example, sequence data where there are well-known databases available. Currently, we do not check for that. So, but that is also something that we would like to address. Yes, of course, if you, for example, have a sequence data and protein structure data, then you should upload those to the right structure databases, and then use BioStudies for creating a very simple entry. Just link to sequence database entry, link to protein database entry, and the title of the dataset. So yes, it is a well, of course, the BioStudies submission process is much more lightweight compared to other databases, and we should be careful not to take away data that should belong elsewhere. I hope that answers the question. So the next question I see: are all submissions done by investigators themselves? Can you also expand the database by adding missing studies that were already published? So no. So the most of currently of the 2 million datasets present in BioStudies, most of these have been automatically acquired by the pipeline that is run in the Europe PMC database team. So they mine text of the publications, they find accession numbers, then they also find the supplementary data files, and then that is the data package that is sent to BioStudies, and we make it available from BioStudies. But as I mentioned at the beginning, it's automatic process, so it's not always foolproof. So it is better that investigators submit the data beforehand, and then there is a single data package that is already hopefully well formed, and so yeah. The next question: is it possible to apply a DUO code to the datasets? Yes, I know about data usage ontology. I have, but we do not yet use that. That's something, another point for us for the to-do list. Thanks. That's a great question. Can you say something about how much BioStudies is used by the community? Well, it is. The usage is growing since we came live 4 or 5 years ago, and as we are migrating, for example, ArrayExpress BioStudies, so ArrayExpress still has more unique visitors per month compared to BioStudies. I think this will raise the awareness of BioStudies in the community. So yes, I do not have concrete figures in my mind at the moment. The next question: could BioStudies used for papers published not in open access mode? And what about copyrights? So yes, it's not limited to open access papers. For non-open access papers, BioStudies will not get these automatically created data packages from Europe PMC, but for data sets prepared by investigators, that can be done, and all the copyright still stays with the data author. How does BioStudies handle personal or GDPR sensitive data? Yeah, so that is something I meant essential. So all the databases at EBI, except one, European Genome Phenome Archive, are open access and are, well, essentially each data set. Well, it can be in a confidential status before the paper is published, for example. But eventually, the data set should become public, totally public, without any restrictions. EGA is an exception, and EGA enables sort of the mode of work where you have data access committees for a particular project. So then to get access to a particular data set, you write your proposal to the data access committee. You get access, and only you get access. So that's not something that BioStudies can do or would do in future. Next question is who can use BioStudies? It's everybody. Well, it's especially for data access. Yeah, as I mentioned, all public data sets are just like that. So you do not need to log in. For data submissions, again, it is we do not have anybody checking the individual accounts. So if you need to share a data set, you can sign up for an account. Yes, and then once you have an account, you can publish your data set. So there is a question about RMA normalization. So BioStudies, it's sort of data file agnostic. So all we do is we take the files from the submitters together with the metadata and annotation, and then we provide the files back to the users, data consumer. So we do not do anything. We do not process data files in any way. So yeah, that's not. I think the thinking in future, as a big. So I mentioned this sort of idea of using BioStudies for a particular project. So I can imagine that in principle it could be possible to think of sort of some plug-in architecture where for a particular data collection we somehow process the data files automatically, but that's not currently in scope, and that's not something that so a question from Katya: once uploaded on BioStudies, can someone apply restricted data labels until publication? Yes, absolutely. So essentially, if your release date is 2 years in future, then that will be a restricted access data set. So only you will be able to access that, and you also get the secret URL that you can give to the journal, and then the journal editor can give it to reviewers if necessary. So they get to see only this particular data set. If you have 3 data sets, then for each of the 3 you get a different secret URL. So you can do that as well. So yes, that's possible. Jupyter Notebook implementation. Yes, so that's great. So it's still very much a work in progress. I think it started around 6 months ago. EBI has a cloud environment. We call it Embassy Cloud, where it's possible to deploy Kubernetes-based environments, and so there we have sort of a pilot implementation that allows, for example, take a Jupyter notebook that you have on GitHub, use the binder system, and then the binder essentially does its magic, and you get your Jupyter notebook running, and then in that running Jupyter notebook you have access to data files in BioStudies, and because these 2 systems sort of run next to each other, there is no network bandwidth problem. So that's perhaps a very sketchy description of what's happening. I think this will be sort of perhaps in the next, I would say, in 6 months or so, it will be advertised, perhaps in the bioimaging context, and then we will also look at sort of more general access for BioStudies. That's a sort of quite vague answer, but watch this space. Thanks everybody for coming and listening.